All right. Now do you see it?
Yes.
Looks good.
By attending this meeting, I am consenting to being included.
Ooh, and it's
Now I-
transcribing what we're saying. I don't know if we really want that, but
I think you can turn off transcription for your own personal
Oh.
viewing.
You can hide it.
Oh,
Okay.
no, transcription has stopped. You're now only recording.
Perfect.
I think perfect. Okay. All right. Well, maybe we'll get going. We're up to 28 people. So, oh, it's still showing the transcription on my chat.
It is. Sorry, I restarted it and tried to hide it because Caleb wanted me to, but if it's still showing on your chat, I will stop it again. Did it stop showing or is it still showing?
It's still showing.
All
Still
right.
showing.
Sorry about that.
And now it says transcription has stopped.
Okay, thank you. All right, so why don't we get started? And so maybe Ruth is going to kick off our presentations, but maybe you could just put up the agenda just.
Uh-huh.
And so, we showed this morning, but we're going to have 3 presentations. The first is going to be Ruth giving an update on the Avian Knowledge Network Colonial Waterbird database. And then Gemma Klukas is going to give a talk on fecal DNA to document common tern diet. We mentioned the Project Poop project this morning. I'm going to give a brief update about that collection effort, and then we're going to kind of open it up for discussion, get some feedback from people, hear about potential suggestions for next year, what we might want to do differently. And then hopefully we can dive into some of the short and long-term objectives of the Forage Fish Working Group. We've really focused the last 2 years on trying to understand what diet information was being collected along the Atlantic coast. And then when we realized outside of kind of the Northeast, there wasn't a lot being done, we implemented the Project Poop project. And so hopefully we can maybe start tackling some other objectives that we've identified in the past. So maybe with that, Ruth will kick it over to you for the AKN update.
Okay. Let's see. Slideshow. Oops. All right. Is everybody? Oh, why am I? Hang on.
Going on a field trip?
There we go.
Rat.
Oh, is that a puppy? I can't see anybody, but I thought I heard some kind of gnurling noises.
That's what it's, maybe it's a baby, but I thought puppy too. So
can everybody see my screen okay?
Yes, it looks good.
All right, excellent, excelente. All right. So yeah, I'm just gonna do a brief update on the development of the Atlantic Flyway Colonial Waterbird Data Management System that is in the AKN or Avian Knowledge Network. And I did present a lot of this stuff last year at this meeting, but I wanted to make sure, we may have new folks. So I apologize if some of this is a repeat for some of you guys. Anyway, admit. And I am not advancing. Gott den Himmel nochmal. I love technology. I'm going to stop and start again. That seemed to work before. Let's try this again. God. Still not advancing. Does anybody have any bright ideas?
No, I feel like I don't know if I use the enter key. I tried the arrows.
I did
Or
all
if
of
I
that.
did-
I'm gonna.
Have you tried the
I'm
mouse
gonna-
button? Could you share your screen? And
I don't have. Yeah, I don't have a mouse. So let me try this again.
yeah, right. You're just sharing your screen, right? Can you do it from there? Even if
Oh,
it's
you mean instead of let me first see if I can get this guy.
Yeah, share
I'm
screen rather than the PowerPoint app. So
Yeah, since you're unverified, the app probably won't work properly. So sharing your screen.
Oh, it won't. Okay. Oh, there we go. Voila!
oh,
Whatever
okay.
happened, who knows?
Glad to be wrong.
I apologize, you guys. All right. So the reason why I've been giving these updates to this group is because SCAW actually adopted this particular objective back in 2017 when it was first formed, and that is to coordinate, facilitate seabird breeding data storage, sharing, and analysis. So I do want to point out that because this is a centralized data management system for all colonial nesting species, it's designed to not only house seabird breeding survey data, but also data collected on long-legged wading birds. So I'm just going to go over some of the progress that's made since last year, and then just kind of lightly step into the weeds and talk a little bit more about the required core fields that serve as the framework for this data management system. So I'm going to start by just providing a little bit of background. So in 2020, the Atlantic Flyway Council funded the integration of colonial waterbird data into the Avian Knowledge Network, or AKN, through a one-year contract with Klamath Bird Observatory, which is a key partner of the AKN. And the phase one deliverables included the compilation of historic data, which dates back as far as the 1800s, along with a couple of more contemporary databases, and then transformed all of these records into more manageable format. Another deliverable was the construction of custom data tables in which data were imported, and then the creation of user interfaces to access the data, and then also establish data sharing levels. And then phase two of this effort, which was completed under a three-year contract between KBO and the Atlantic Flyway Council, we really started to get into the weeds and involved state, provincial, and non-governmental waterbird biologists and data managers. And the reason why is because it was very apparent that the historic and more recent data that were uploaded into the AKN was messy, and the existing structure was really clunky and didn't really reflect the needs of states and provinces. So KBO worked with two teams of biologists and data managers over the 2 years. And together we basically overhauled the existing database structure. And that work involved identifying and defining core fields and identifying and defining values for each of those core fields to ensure that they are relevant to all states and provinces in the flyway. And then we also developed a definition for sites and colonies. And then lastly, finalized state and provincial species lists. Oh, and one thing I forgot to mention. So I bolded a couple of these bullets because I'm going to talk about them in a bit, just to give you a sense of what the database structure looks like. And now we're in the final phase, or phase 3 of this project, which again is being funded by the Atlantic Flyway Council, and the completion date is February 2026. And the phase 3 deliverables, which also requires input from Atlantic Flyway colonial waterbird biologists, include creating custom database for each state and province. And these databases will, in addition to the core fields, will also include additional state or provincial based data fields that are necessary for informing more local colonial water management decisions. And I'm happy to announce one of the biggest, a lot of progress has been made in getting these databases set up. I think most states have their databases set up in the AKN, and they're currently in the process of entering their 2023/2024 colonial waterbird survey data. So that's pretty exciting. And so now that, you know, these data spaces are getting set up, the next step will be staff from Point Blue Conservation Science, who are the AKN hardware engineers. They're going to build an overarching database structure called the Atlantic Flyway Program Enterprise that will facilitate data sharing, access, and analyses across states and provinces. And hopefully other flyways will adopt this as well. And so eventually, you know, in a perfect world, you know, analyses can be done across flyways, so almost nationwide, which would be pretty cool. We'll see if that happens. All right, and some of the remaining deliverables or tasks under this this contract will be the development of mapping, analytical, and other interactive decision support tools, which is going to be critical, and developing an application programming interface and customized data outputs that will allow state and provincial databases to feed directly into these interactive tools. So that's going to allow, you know, hopefully, you know, make it easy for people to pull up these data, extract the data as they see fit to do the analyses that they wish to do. And it could be at all spatial scales, which will be nice. So once this work gets done, it should be ready for this database should be ready for prime time. And the data management system is going to be managed and maintained by the Atlantic Flyway non-game migratory bird tech section and the Flyways Waterbird Committee. All right, and so now I'm going to get a little bit into the weeds and talk about the core fields. And core fields are common to all state and provincial databases within the Atlantic Flyway Program Enterprise, and they contain standardized data that can be extracted by users from whatever state or provincial databases they choose, and then subsequently be queried, analyzed, and visualized across multiple spatial scales. And these fields were developed for the purpose of tracking population trends, shifts in distribution, and habitat use over time across broad geographic scales. So here is a sample data sheet that contains all the core fields and all the possible values that we identified for each of these fields. And I know it looks pretty daunting, but you have to keep in mind that these long lists of values were developed for each of these core fields were developed to meet the needs of all the Atlantic Flyway states, and which you can imagine encompasses a whole lot of different site and habitat types. So the fields that are in this upper portion of the data sheet are what we call site level fields. And so they include your typical stuff like date, you know, start time, observer information, that sort of thing, colony name, colony lat long. What's new is the site name, site landmass, and site water body. And so the groups that worked, you know, to develop these core fields, decided it was really important that each database identify sites that are sort of permanent features on the landscape. And those sites are going to be monitored over time. So each site is going to get a unique ID number or name, and you know, the site can either support one or more colonies. There are going to be some years where the colonies don't show up there. We know that, you know, colonies are very ephemeral. They flit around and that sort of thing. But we wanted some permanent feature that can be tracked over the long term until that site completely disappears from the landscape, right? So once that happens, then that site name can be removed from the state or province's database. And it could be anything from an island, you know, an impoundment, a structure in the water, anything that's sort of a permanent feature on the landscape. And the site landmass, this guy here, that basically describes or identifies the type of landmass or overarching land feature of the site. So, and unfortunately, I don't have the codes for those up here, but basically it could be, you know, like a offshore island, a barrier island, a segment of a mainland shoreline or river shoreline, and you know, each individual state has to kind of figure out the boundaries of the site. Could be a wetland, an impoundment. It could even be a structure in a water. You know, so just whatever's that permanent feature. And then the site water body is it basically identifies the water body that's closest or adjacent to the site. And that could be the ocean, an ocean inlet, a tidal bay or sound, a river. You know, we have a long list of values for those too. And I think we got those all covered as well. So those are kind of the site level variables. Oh, and one other thing is the groups also decided not to add weather variables in the database, just because, you know, a lot of that stuff really never gets used. And so instead we decided to just ask the question whether weather affected the quality of the count or not, and left it at that. Okay, so then getting to the species levels, variables. You know, the first two columns are just, you know, the species, obviously, and your raw count. Units counted here are, and here we're, and on all of these all of these that I'm going to describe from now on, they do have the codes in the boxes. So anyway, it's just, you can just kind of read through the units counted. It goes anywhere from adults to active nests to breeding pairs, right? It has, I think, every conceivable unit you could count. The count type is whether the count was a complete census of the colony, whether all active nests were counted, or just a portion of the nests were counted in a colony, whether it was an approximate estimate, a flush count, those kinds of things. And the percent of the colony survey only pertains to count types that were partial or incomplete, right? And then count method is, you know, the method, basically the platform from which the count was made. So from either within the colony, on the ground, from the perimeter, from a boat, a car, you know, the whole gambit, drones, satellite photos. Yeah, we try to catch it all. The reproductive stage is basically a way to kind of define the quality of the count, and it's just a visual estimation of the reproductive stage of the colony itself. You know, when you go into a colony, you know, some birds are still going to be incubating, some birds will have chicks, that sort of thing, and it's just a visual estimate, and you're just recording percentages of the number of birds that were either, and here are the reproductive codes for those, whether they were incubating, whether they were still nest building, you know, if there were unfledged young in the nest, what have you. And you just kind of estimate. So like 20% of the birds were incubating, you know, that kind of thing. And it has to add up to 100. And then the data quality, that was another way to sort of characterize the quality of the count. And here are the codes for those: high, medium, and low. And basically the data quality takes into account the site conditions, whether the weather was good, the detectability of the birds was high, the date was correct. It was, at a good point in time. We always like to try to catch them at peak incubation. And if all those conditions were met, then the quality was high. And then we kind of go beyond, medium, everything was adequate; low, the conditions were pretty suboptimal. And then we also came up with habitat type codes, and that took a while to go through. Basically, this defines sort of the habitat types in the context. I'm sorry, my dog is barking. I gotta go let him out. I'll be right back. Good. Sorry about that. They always pick the best time. Anyway, we came up with habitat type in the context of further dividing the space the colony inhabits into sort of management units. And those may change over time, right? So we also realized too that a colony can encompass different habitat types. So there's primary, secondary, and tertiary. And here you can see all of the different habitat type codes: forested, shrub dominated, could be swamp, salt marsh, freshwater marsh. It could be rock outcrop, rooftop. That's all those sorts of things. And then we also further defined habitat type by classifying it into natural, restored, managed, created, and then mix of natural, restored, managed, a mix of natural, created, and opportunistic. And so people know, like, a natural site is a site that's never been modified. There's been no restoration done on it. Restored, managed is a site that was modified in some way, shape, or form, either for birds or for enhancing coastal resiliency, those kinds of things, but it had been modified and is being managed. And created, of course, as you go out and you build a whole new island, so those sorts of things. Again, it's just a way of kind of defining or identifying sort of where these birds are nesting over time. And then lastly, we have the nest substrate codes, and that is the substrate that the birds are actually nesting on. So you could be trees, it could be sand, shell, a rock or a boulder, cobble, again, the whole gambit of of every type of surface that these birds can nest on. So these are pretty much, this is kind of setting up the entire structure of these databases. They're all common across states and provinces. And so it makes it, it's about as standardized as we could get it. And then, like I mentioned earlier, individual states and provinces could add their own fields that they need for their own management purposes. So that is all I have. So I'm going to stop sharing. And I don't know, I can probably take a question or two, maybe. Does anyone have any questions? Yeah, Yvonne.
Hi, looks like a great effort, and it's good to see an update on it. You mentioned data sharing levels at the beginning. How did you manage that, and what are the levels for it?
So, sorry. So the AKN actually has their own data sharing levels. There's biologists, project leader, that type of thing. And what we decided was we would go with that format and have the individual managers of the databases, the state and provincial database database managers select those people that can access the data and at what level. So with it through the AKN, every time a person requests information from that program enterprise and from your database, if it's your database, you're going to get dinged, you're going to get an email, and you will have to respond to that email saying that you give permission or not, or deny permission. Does that make sense?
Yeah, okay.
And you know, there is, I'm sorry, I just don't have the list of the actual sharing levels in front of me. I apologize. I can get that for you though, if you want that. And I also can, anyone who wants, you know, the sample data sheet. And then we also developed a cheat sheet that describes all of the variables in detail. I can also provide that information as well.
Thank you. Yeah. If nobody has another question, I have another one.
Okay.
So tomorrow I'm presenting on the effort we're doing in the Gulf of Mexico, which is
Yes.
different, but we had a lot of historical data, and unfortunately they don't, the units being measured don't match across providers. Sometimes they're just nests, sometimes they're just adults or breeding pairs. But you do mention that database users could do analysis. So how can they do analysis? Sorry, in your case, do the units match across a region, for example, a flyway?
So I'm glad you asked that question. Okay, so we decided what the common metric is going to be is nests, number of nests, right? And so when we first started with this project, I was amazed what people were counting, right? So it is up to the data manager to convert the units counted into number of nests. And that is going to be and it's not included in this data sheet, sorry, because it was just a data sheet. It was the only way I could figure out how to present all this stuff in one screen.
Yeah.
So anyway, but yeah, the common metric will be nests, and nests will get each data manager is going to have to convert that units counted into nests. Does that make sense?
But yeah, it does. And I guess there will be a way for them to record how they converted.
Correct.
Right.
Yes.
Okay. Great. Thank you.
Yep. All right. All right. I think we'll move on. And Gemma, if you can go ahead and share your screen.
Yeah, absolutely.
Like there is a question in the chat.
Oh, sorry.
Oh, I tried to answer it. There was a question about whether you could designate artificial or human-made habitat versus natural. And I wrote yes, that you can. Maybe, hopefully that's right. I feel like we talked about that quite a bit.
Yes.
And there has been a lot of created habitat, so I feel like we tried to capture that.
Yeah, you can. That's correct. And what's cool about that is that, you know, for the first time, sort of on a broad scale, it'll be able to see sort of the success of created habitats, right, over time, which is kind of cool.
All right. Thank you, Ruth. And maybe we'll move on to Gemma Klukas talking about using fecal DNA to understand common tern diet. And then hopefully we can talk about all of this in the latter half of this session.
Cool, thank you. So for anyone that doesn't know me, I'm a research associate at the Cornell Lab. And I just want to start by thanking everyone who's been involved in this over the last sort of 3 to 7 years. There's a lot of samples that have been collected to go into this, and so thank you everyone for their involvement. So we've been focusing on common terns as an indicator of forage fish availability in and around the Gulf of Maine, because they're the most widespread breeding seabird, and so we've been sampling them at all of the filled colony circles here. And we've been sampling each of these 13 different colonies for anything between 3 and 7 years. Common terns are also generalist in their diet, and so we don't think they're being particularly picky in what they're eating, and they're easy to sample, so we don't have to go diving under rocks and things to access them. So we can sample chicks opportunistically during regular banding and productivity checks. This is a photo that we didn't stage very well. Normally we would clean our scales very well between weighing different chicks so that we could sample off clean surfaces. We've also found putting them in paper sandwich bags provides a nice clean surface to sample from, but we'll also sample from our hands or our clothing, assuming those are quite clean when they poop on us. And then the adults we can sample as well. So I've just found that walking through the colony with a trash bag on my hat provides, again, a nice clean surface to collect those adult samples from. And so the method is fairly kind of straightforward. Theoretically, you just collect samples from both adults and chicks. We extract DNA from those fecal samples and then sequence what are known as barcoding genes. So they work like barcodes in the grocery store. We sequence the gene and then match it to a reference database in order to determine exactly which fish the birds have been feeding on. And we are just focusing on the fish component of the diet and ignoring invertebrates because they make up such a smaller proportion of the diet in most years. The lab methods are quite involved. Each of these steps takes multiple hours to do, and so I'm really, really reliant on a fantastic team of undergrads that I work with. So they've been trained in all of the lab methods, and we pay them to come in and work on these samples every week. We do have a liquid handling robot, which helps sort of reduce some of the manual pipetting that we have to do and helps reduce human error. And we've just secured funding to buy a second one of these robots. So that will help increase capacity a little bit over the coming year. Where I spend a lot of time rather than in the lab itself, I spend more time on the bioinformatics and particularly in manually checking all of the species assignments. So I go through all the sequences, I check, is the fish that it's been identified as actually found in the Gulf of Maine or as we expand, is it found on the East Coast? Are there other closely related species with a very similar DNA sequence that I think it could be? Therefore, am I confident in making a species level assignment, or is this better sort of a genus level assignment? So I spend a lot of time manually checking all that data. And so that's all just to say that although the field component is relatively straightforward, the lab work still takes a lot of time. And so I just appreciate patience with everyone I'm working with in getting the data back to you, because it is quite a lot of work. Liz Craig and I have spent quite a lot of time working on validating the method. So we really wanted to show whether we could use the dietary DNA, not just to list the species in the diet, but accurately estimate the proportions of those different fish in the diet. And if we could do this, how many samples do we need to be able to collect to do that? So we spent a lot of time observing out at shoals the proportions of different fish that were being fed to the chicks, and then comparing that data to the fecal samples that we were collecting at the same time. And so we're comparing at the colony level, not at the individual chick level, but we've managed to show that both in common terns out at shoals and in Atlantic puffins, thanks to Will Kennelly and the crew at Matinicus Rock, we've shown that we do get this clear, strong, almost one-to-one relationship between the biomass that the chicks are being fed of each of the different fish species and the relative proportion of DNA sequences that we get back from those species. So we do think we can use this method to track changes in proportions of fish in the diets of these birds over time. And then that second question of how many samples do we need to accurately estimate these proportions? We did this with Andrew Stillman, who's a colleague at the Cornell lab with me, and he simulated data sets ranging from 10 to 40 samples collected per year. So as if we collected just 10 samples per year up to 40. We then compared those simulated data sets to the actual biomass that we'd observed being fed to the birds. And we found that the error between those data sets declined and stabilized around 35 to 40 samples per year. So when I'm sending out sampling kits and asking people to collect samples, you know, trying to hit that upper limit of 40 samples for chicks and 40 samples for adults is really important so that we can accurately estimate the proportions of fish in the diets of these birds. So to go into some results from those colonies in and around the Gulf of Maine, as I said, we've been collecting from 13 different colonies over the past 3 to 7 years. And we've been able to detect many more species than ever before. So I think I've processed over 5,000 samples now, and we've detected 83 different fish taxa. But the vast majority of these only turn up in relatively small proportions in the diet. And actually, the vast majority of the diet of the terns is made up of relatively few different species. So Atlantic herring makes up about 50% of the diets of the terns in the Gulf of Maine and surrounding colonies, and then sand lance makes up about 20% of their diet. We have white hake at about 10% of the diet. And so we know from visual observations that hake can be an important diet component. And now we can say from the fecal DNA that actually the majority of that hake that they're bringing in is white hake, with a much smaller proportion of silver hake, and then an even smaller proportion of red hake, which is in much less abundance. Similarly, we can now distinguish the river herring from the Atlantic herring. So river herring only makes up about 5% of the diet in total, but as I'll show in a bit, it depends on which colony we're talking about in terms of how the proportion of river herring versus Atlantic herring varies. We then also see quite a large proportion, 5% of the diet is four-beard rockling. Probably that's mostly being identified as hake. They look quite similar in visual observations, but actually rockling are quite important in the diet. We then got the gaddids, pollock, haddock. We also see Atlantic cod and Atlantic tomcod in smaller proportions. We've got mackerel, we've got butterfish, so those larger chicks, and the adults are able to swallow some of the smaller butterfish. And then, as I said, some silver hake as well. Going back to the bigger table, we're able to distinguish 4 different species of sticklebacks. We've got 3-spine, black spotted, 9-spine, and another one that didn't have an exact match. So I just gave it a genus level assignment there. We've got 5 different species of flounder, so summer, smallmouth, witch, and windowpane, and then another one that I couldn't identify at species level exactly. There's some of the weirder things in here like goosefish and lumpfish and pufferfish. One of the interesting ones was a lionfish. I think this was back in 2018 at Shoals. One of our incubating birds had lionfish in its diet. So a really strong indicator of that warming trend in the Gulf of Maine if even juvenile lionfish are potentially being swept in on the Gulf Stream now. And then the most weird one out of all of them was this ocean sunfish that came up. Obviously, sunfish get absolutely massive and no tern is going to be feeding on an adult, but they have to spawn. And it's not really well known where these sunfish do actually spawn, but if the terns are feeding on larval sunfish, then I guess it must be somewhere in or close to the Gulf of Maine where these larvae end up as tern prey. We can then ask whether adult and chick diets differ, and the answer is not hugely. So in this PCA on the right Each point is a colony where we've sampled, and the adults are in the green and then the chicks are in the orange. The sample size is represented by the size of the point, and so anything less than 40, like these ones, we should take with a pinch of salt. But you can see there's a large overlap in general between the adult and chick diets. And then going species by species, we can see there's a little more Atlantic herring in the adult diets relative to the chicks. This is likely a timing thing. Our adult diets or adult samples span June and July, whereas the chick samples are concentrated in July. And we often see in the summer, as things warm up, herring become less available as time goes on throughout the summer. And so we see slightly more herring in those adult diets. There's a little more sandlance in the chick diets, quite a lot more white hake in chick diets compared to adults. And again, this is probably because of when we're sampling. We think those white hake become available later in the summer when we're mostly sampling chicks. And, you know, going down, the proportions are fairly similar as we get to those more or less common species. We can then dig deeper and look at how diet varies by year, by location, and between adults and chicks. There's a lot of data here, and so to kind of simplify visualizing it, again, I'm using principal components analysis, and I'll just take you through how these plots work. And so on the left-hand side of the plot are the colonies ordered latitudinally from north to south. These ones above the dotted line are all within the Gulf of Maine. And then these 4 colonies south of the dotted line are down here, south of Cape Cod. Adult diets are going to be shown on the left compared to chicks on the right. We've got year on the x-axis, and then again the sample size will be shown by the size of the point. And then the color is going to represent position along one of the axes of the PCA. So starting with PC1, which explains the vast majority of the variation in the data set, this really separates diets dominated by Atlantic herring, which are shown in pink, and sandlance in yellow. And so you can see this big kind of stark difference between the colonies in the Gulf of Maine, which really are dominated by Atlantic herring in the diet, versus those found south of Cape Cod, where sandlance really dominates those diets. And that's not unexpected. You know, we know there's a lot more sandy habitat available down here. Water temperatures tend to be warmer as well. But this is the kind of the big pattern in this data set that we see. The other thing to point out is that herring was most abundant in the diet in 2019. So we see sort of the darkest pink in 2019 here and here. And 2019 was the coolest year out of these years that we've been sampling. So we didn't see any periods of extended heat wave conditions in 2019. And so that seems to indicate that, you know, that was a good year for herring. We do see some sandlance in diets in the Gulf of Maine, so particularly around Stratton. We have sandy habitats available there, explaining why we see a bit more sandlance at Stratton at that colony. Moving on to PC2, this really picks out the influence of white hake in the diet. As I said before, white hake is much more abundant in the chick diets than in the adult diets. And we think, again, that's because of the timing of this sampling. And so white hake probably becomes more available later in the summer when we're sampling those chicks, compared to the adult diets where we're sampling often a little earlier in the season. And the distribution of hake in the diet is patchy in both space and time. So we see hake again down at Stratton and seal in some years, some at Petit Manan, although we don't have many samples here. But this is kind of typical of these species that go through boom and bust years. You know, sometimes hake will be very available, sometimes less so. And so we can really see that in the diets of the birds. And then moving to PC3, this picks out the influence of river herring in the diet. So here with river herring, I'm grouping the blueback herring and the alewives together because genetically they're very similar, and I don't think we can reliably distinguish them from one another. So grouping them, we do again see they're an important diet component, but only in some colonies in some years. So at White and Seavey, they seem to make up quite a big proportion of the diet, although not in 2019 when Atlantic herring was more available. And then we also see quite a lot of river herring at Jenny and Pond Island. So again, we have kind of big river estuaries coming in both of these areas, likely providing good habitat for river herring. And so as sort of efforts increase to restore these river herring populations, we might start to see more river herring in the diets of these birds. And so being able to distinguish them from the Atlantic herring is really an advantage of using this fecal DNA method. And I'm actually talking with Sean Hardison, who's doing the Atlantic herring or developing the Atlantic herring index from the seabird data from the Gulf of Maine. And he's going to do a sensitivity analysis to see whether these proportions of river herring that we're picking up in the fecal DNA will actually influence that index of Atlantic herring, because they're just working so far with the observational data where you can't distinguish the Atlantic herring from the river herring, but we can add that component now with the fecal DNA. And so Linda is going to talk about how we've next expanded kind of the sampling to cover terns up and down the East Coast, and including black skimmers as well, all the way down to Florida. And I think I missed Mississippi off here as well. Sorry about that. But just kind of to summarize, the dietary DNA allows us to collect this really high taxonomic resolution data that does reflect the relative biomass in the diets of the birds. We can easily compare adult and chick diets, assuming we can get samples from both. And we're now scaling up the sampling so that we can look at these ecosystem scale patterns and hopefully, use this data for forage fish management ultimately. So thank you everyone who has been involved so far and continues to be involved, especially all of the field crews who have collected a lot of samples and been pooped on a lot. And I think we probably have time for a few questions. I will stop sharing if I can figure out how.
Sure. It looks like Carolyn has a question for you.
Go ahead.
Thanks, Gemma. So incredibly appreciative of the work you're doing. It's really amazing. So thank you so much for that. I wanted to ask about the potential to include some non-fish in the analyses. I think we might be seeing increasingly significant proportions of squid in the diet. Maybe because fish, some of the reliable fish are declining. Is there a possibility to include that in the future? And maybe there are some other colonies that are seeing more invertebrates as time goes on as well. We do see shrimp as well. I'm not sure. It still seems to be relatively minor, but we are seeing more and more squid as far as I can estimate.
Yeah, we can do an extra PCR that will target, it's kind of a more generalist one, so it would pick up squid and also the invertebrates, but we wouldn't be able to get to species level. But assuming that doesn't matter as much, then yeah, we can do that if you feel it's important to start looking beyond fish. That one is more expensive because it picks up so many different taxa, because it's just kind of general eukaryotic, or it targets general eukaryotes. We have to sequence a little more deeply in order to try and kind of detect everything in the diet. And so it does come out a bit more expensive. but we can definitely think about including that in the future.
I mean, maybe it's something that at first you could deploy on a more limited scale than the regular analysis, since it's more expensive and probably more time consuming as well to add that analysis, and then see if it warrants being deployed more broadly.
Yeah, then we could start with bird and ram and then see how it goes.
It's good. Thanks, John.
Tasha?
Hi. Thanks so much for your talk and for such amazing visualization of really complex data. It's not easy to do and you make it look easy. So thanks again. And yeah, I was going to ask about inverts. I was curious. It sounds like you hadn't run on any of those, but I was also curious whether you had thought a little bit about it all during this process about, I don't know how often you've seen like the fish scraps brought back, but we see them quite a bit kind of in bouts on at least Petite Manan, and was just looking at some other, data on C to N ratios, and the predicted lipid content of those samples is like above 80% or something like that based on the C to N ratio. So I'm just curious if you had any thoughts on, based on any weird results that you saw, that you might think, what they're bringing back as as those scraps.
What scraps, are you able to identify what they're bringing back in those, or because I mean with the fecal DNA we can't really tell. Yeah,
I was just curious if you had seen anything weird that you were like, oh, this must be fish, like bait or something, rather than it looks just like kind of chunks of fat. I know Linda mentioned it could be like pig skin that sometimes fat's attached to, but I don't know. I was just curious if you saw anything weird in your results that you were like, thought was maybe related to bait in any way.
But what about redfish, Gemma? Like some people are using redfish for bait, and we had said that terns likely wouldn't be able to catch redfish on their own, but they may be getting it as discarded bait.
Yeah, we do see redfish coming up. So that could be.
Yep. But in Maine, quite a few lobstermen are using pig hide as a bait because it's getting more challenging to get herring or expensive. And so, yeah, you can get 5-gallon buckets of pig hide. Yum. As bait.
Yeah. That reminds me, we did have pig come up in, it was like our first year of sampling at Shoals, and I think we put it down to the fact that we'd put our samples in the freezer where there was maybe bacon before, but it actually maybe it was terns foraging on bait. So that has come up. So with the fish, if they're using locally caught fish bait, then it would be hard to distinguish. But if we do start doing more of the kind of general eukaryotic sequencing, then we might see some of those more weird scraps coming in.
Thank you so much again.
All right, maybe we'll move on to my quick update on Project Poop. But again, we'll have lots of opportunity to talk with Gemma and get her input on maybe next steps. So let me turn that on. Share screen.
And while you're doing that, Linda, I just want to, I think we sent out an email to all the folks that did participate in Project Poop in 2024. And we do plan on holding another sort of pre-season meeting in February. And so we'll certainly have Gemma join us, and questions can be asked at that time as well.
Okay. Can you see the Project Poop update slide? Okay. All right. So we've heard a little bit about this project already, but in 2023, we did a survey of colony managers to try to understand what diet information was being collected at the colonies. And we realized that outside the Northeast, a lot of colony managers just didn't have the capacity to collect detailed diet information. So we wanted to try to figure out a way that we could start getting some information from additional colonies up and down the East Coast without putting undue burden on the colony managers. And many of us in the Gulf of Maine had already been working with GEMA for a number of years. And so it seemed like a great opportunity to extend the fecal DNA work. So our goal or objective for the Project POOP was to develop a coordinated and standardized monitoring framework and database for seabird diets across species along the US, Atlantic coast, for landscape and climate scale analysis. And obviously we're also including Canada, so we need to change the way that's worded. So again, this was a regional effort implemented in 2024. And again, just trying to get a basic understanding of what common terns and black skimmer were eating. And Ruth identified those were 2 priority 1 species in that colonial waterbird survey effort. And so we focused in on those 2 species. We're going to use fecal DNA to document the seabird diet, and samples were submitted to GEMA. And so last year we kind of viewed that as a pilot project. In this fall, Ruth developed a survey that we sent out to people that said that they were interested in participating. We got 24 responses. 17 managers or researchers indicated that they had collected samples. Samples were collected from 12 states, going all the way from Maine down to Mississippi, and 2 provinces, New Brunswick and Nova Scotia. But the more interesting part is this table. So it identifies the number of samples collected for each species in each either province or state, and then the number of colonies that were sampled are in parentheses. So I was shocked to see how well everybody did. We got over 1,300 common tern samples and 475 black skimmer samples. So almost 1,800 samples collected across this really large geographic range in the very first year of this project. So sorry to GEMA for having to analyze all these samples, but I think this is just a great example of what we can do when we work together. And hopefully the work that we've been doing in the Northeast, where we're starting to see some of our diet information included in some of the fisheries management plans or the ecosystem reports, hopefully, we'll have a little bit more information when we try to engage with fisheries managers about what our seabirds are eating and how they might help to monitor what's going on with forage fish. Going back to the survey that Ruth sent out to people, most respondents thought it was a valuable effort and think they can sample colonies again, the same colonies again in 2025. We also heard from Georgia and Pennsylvania that they will be able to collect samples next year. They weren't able to do it this year, but they will be ready in 2025. And about half the respondents thought they might actually be able to sample additional colonies next year too, so that would be great. We also asked people to identify potential challenges that they encountered. Despite it seeming like seabird chicks poop nonstop, sometimes when you're trying to get a poop from them, then they don't. So we're going to offer some additional suggestions for helping people collect samples next year. It can be challenging to collect samples from adults, particularly if it's in a mixed colony, because you don't know who's responsible for the poop that's raining down on you. So that's one of the things that we deal with up here in Maine. And so hopefully we can help folks figure that out as well. And we had also asked people to identify the age of the chick. And a lot of that comes from many of the samples we collect in Maine are in our fenced productivity plots. And so we know exactly how old those birds are, but there are some resources that we can share with people to help identify chicks. And then we're thinking you might be able to just group it to like, it's less than a week old, less than 2 weeks old, less than 3 weeks old. So I think we can work together to try to help clarify what information we really need and to help people identify the specific age of the chick. So we didn't hear anything, I don't think anything too out of the norm for problems. It was, some logistical challenges and then, like, oh, we had plastic on our hat and the poop didn't stay on it or something. So I think there's definitely room for improvement on guidance and support documentation. And that's all I had for the Project POOP part of things. Let me stop sharing right now. So that was very quick, but I don't know if anybody has any questions about that project in particular for hopefully Gemma and not me. Or if we want to jump into a general discussion on kind of next steps for that effort. I know in the chat, Jeff Spendelow had suggested it would be great if we could collect some samples from staging birds, and he thought that that would be realistic to do. I think, you know, there's a couple of logistical challenges, like what is Gemma's capacity to collect, if we collect additional samples to analyze them. And we were saying, you know, maybe we really need to think about kind of a regional grant opportunity. We've talked about this in the past, but I think now that we've shown we have a year of data collection behind us, it might be more appealing for like a multi-state wildlife grant or something to show that kind of the power of this collaborative effort. And then maybe we could help fund additional lab support for Gemma, or help fund, you know, NGOs and state agencies that don't have the capacity to fund these samples. It looks like it was a little bit less than $10 a sample, Gemma, is what it's costing.
Yeah, it's gonna go up by, unfortunately, at least $2 per sample, because UNH, where I do my sequencing, have just jumped their prices by $2 per sample. We're still working out our prices for next year, but it is gonna go up, unfortunately.
Yeah, but still relatively inexpensive.
Yeah, I just heard that if you do this at a commercial place, they're charging $100 to $150 per sample. So, yeah, it's quite a big difference.
Yeah. I just, the main chapter of the Wildlife Society just had a session on eDNA, and they were saying, yeah, like maybe $200 a sample, and everyone thought that was cheap. And I was like, holy smokes, you know, there's no way we'd be able to fund that. So, so I think, you know, hopefully we can work together maybe to develop a proposal to get additional funding. But I don't know if anyone else had questions or comments about the project, any challenges that they didn't reply to in the survey that they'd like us to know about, or additional support that the project coordinators could provide in the future. I know, Sarah.
Thank you. Thank you for that update, Linda, and Gemma, your earlier talk was fantastic. I apologize if I should know the answer to this question, but so is there money for Gemma to sort of do the analysis and writing, or is that something that you're hoping would be the next funding phase, is to have someone that really would pull this, you know, multi-state data together?
What would really help would be actually money for a technician who could manage all of the lab work. My salary is covered at Cornell, so I don't need to find money for that, but having someone who could actually manage the lab work and kind of speed up that would really be useful. So yeah, finding salary for a technician would be my biggest priority. Dawn, did you have a question?
Yeah, I'll just jump in on the thought about kind of a large geographic scale funding pitch. And I think that could be really powerful if focused on a very specific question, and specifically thinking about an applied question like informing Atlantic herring management. I think building a proposal around, you know, that specific application, which incorporates, you know, fecal DNA work, probably also though incorporates somebody to do more wrangling of the observational data, and make that just easier for the herring stock assessment team to use, would be good. And I think kind of a big multi-state grant that is compiling data that we're really interested in, it might not be as competitive without the application being a big part of it. But great work, Gemma. Really excited to see this update.
Dawn, can I ask you a question? I do think it would be helpful if we did have like a direct application of the data. But do you think for the herring plan, and I'm gonna, show my ignorance here, it seems like a lot of the colony managers, at least in the Gulf of Maine, were already, putting our observational data into the provisioning database that we share. Do you think
Yeah.
that that's not sufficient for what the Northeast Fisheries Science Center needs? Or I don't know
I
how big of an issue like herring is in the mid-Atlantic. That's the part I have no idea about.
Yeah, I think kind of the visibility I've had of the process with John DeRoba and Sean is that some data management prior to it getting into the model would be useful, and also to like ensure that the powers that be at the New England Council and the people who will have to buy in to a seabird-based measure of recruitment will know that this will continue, that it's on a solid foundation. I mean, we collect the data, but communicating that and having somebody, a face that can organize those data and commit to feeding that into the NOAA process on an ongoing basis is as big a hurdle as just having the data. So there's that. But I'm really struck by your analysis, Gemma, at the colony level on this split between Atlantic herring and river herring. And just thinking about the Amendment 10 process right now for Atlantic herring, I think the biggest leverage point for any real change in herring management right now is around river herring conservation. There's just so much advocacy around that currently at the council that informing that process and spatially explicitly as you're starting to do, Gemma, could be a really valuable contribution. And both the data you've already got, but some additional data, and thinking about filling some gaps, making sure we're monitoring all of the colonies in the New York Bight where concern is most significant for river herring, like in the Connecticut River and places like that. So I think really digging into how the data are applied is likely key for a bigger picture funding pitch, I imagine.
That's great. Thank you.
Yeah, those are great. Thank you. Ruth, you have a question or a comment?
Yeah, well, a comment, and basically Sarah Carpenter in her chat just mentioned this. And this discussion, this little discussion here is so exciting because in all likelihood, the Menhaden is sort of a similar issue in the mid-Atlantic. And I think if we did develop sort of a large scale sort of proposal, I think we can probably add Menhaden to the equation. It too is sort of a high priority issue, in our area. So yeah, I think this is the perfect tack to take in developing a multi-state proposal for sure. Yeah,
And in
Sarah, I
Maine-
don't know if you want to.
I was just going to say, in Maine, we're starting to see large amounts of Menhaden coming in,
Yeah,
in massive bait balls,
yeah.
where just in the last couple of years. And so I'm guessing we're going to start seeing that show up more in the diet of the terns as well. But that would be nice to have better geographic coverage, like to maybe focus on those two species.
Mhm.
And something we do want to talk about in this session is, how do we interact better and engage better with the fisheries management, whether it's the councils, NOAA. We talk about
Yeah.
it, but we're a bunch of seabird people. If Michelle Staudinger on the call right now. It'd be great to hear her thoughts on that. And she's sitting on I think it's the science team for Atlantic herring. So that's fantastic. And she can help represent seabirds. But, you know, we need to engage better with the mid-Atlantic and the Southeast.
Right.
And so, yeah, there's still,
well, it'll
there's
be
lots to do.
interesting to see. It'll be interesting to see from this, from the data collected this year, the samples, whether menhaden does show up in common terns and skimmers. I would imagine they do, but we definitely know royal terns, you know, feed on menhaden, those sorts of things. So it also may give an opportunity to expand the species list as well. And I don't want to give Gemma a heart attack about making this so huge, but anyway, this is really exciting. And I think, yeah, we should engage the Atlantic States Marine Fisheries Commission as well. They're very interested in doing ecosystem type management. And Sarah, you have your hand up, so go ahead.
Well, first, I'm glad you didn't get mad at me for bringing up menhaden, but
No.
but yes. So I think the other thing I was just going to add in that I was thinking royal terns, Don, when I was thinking about menhaden in our region, and we did send Gemma, no rush, Gemma. We did send her samples, so we are collecting poo samples from royal terns, and it's a very it's getting to be a very heated issue in the Mid-Atlantic, and I think that we could also bring in some fisheries experts to a multi-state from Virginia Tech, from VIMS. We could, you know, I think we should come at this from a multidisciplinary approach.
Yeah. Michelle, did you want to chime in on the bigger issue of better coordination and engagement with fisheries?
Yeah, so I think the Amendment 10 process, I thought that the input that we provided to the scoping work for Amendment 10, I think it was very effective. I was pretty engaged in that process. I went to one of the in-person meetings and I participated in some of the online meetings, and I helped lead a letter that we put in on behalf of the Forage Fish Working Group. And the report that was released after the scoping for Amendment 10 had a lot of, I saw elements of the letter that we put in, as well as the letter that I believe Don helped lead from Audubon. There were recurring themes that went beyond seabirds and overlapped with other marine predators of a lot of the same forage fish that the birds are consuming. So I felt like our message was heard. And if you actually read through this really, really long document that the council released after the scoping process, you could see our comments in it. And then in the following management meetings that were held in the months thereafter, they talked about seabirds. They talked about this more ecosystem-based approach around Atlantic herring management. And I think Linda showed some of my slides this morning that reported that the fishery is sort of in a soft closure right now, but it is not being actively being overfished. There aren't enough fish out there right now to make it a viable fishery. And that points to more environmental problems that are controlling the populations. And that is definitely something that I think we all should be keeping an eye out and trying to think about how we can provide information to fisheries management by helping them understand where recruitment is happening, and that's where the fish are finding those small individual fish, which is an area of uncertainty in the current management plan. But also, if there's ways for us to collect fine-scale environmental data at colonies, I think that could be something else that could be helpful to understand the habitat piece that may be controlling recruitment and distribution of those early ages. So I'm continuing to sit on the Science and Statistical Committee for the New England Fishery Management Council for the next several years, and I was just appointed to the Ecosystem and Climate Committee as well. So I'm listening. I think I straddle both the fisheries world and the seabird world. And so I'm listening and I'm trying to reflect what I hear at the seabird meetings, at those fishery council meetings, and also translate and bring back the information that I hear at the fisheries meetings here. I think Don and others have done a lot of work to make seabirds part of the state of the ecosystem reports that come out annually. And now we're hearing seabirds being taken into consideration in terms of herring specific management. So I think we're making a lot of progress and we're in all the right places, and just staying plugged in and continuing to do what we're doing and looking for new opportunities, I think will be really helpful. And to the point about menhaden that's been brought up, I've been talking to folks at the Maine Department of Marine Resources about the surge in menhaden that has been happening in the Gulf of Maine, and the lead biologist that's in charge of menhaden and it's spanning the entire, he's the rep on the Atlantic States Marine Fisheries Commission as well. He's basically said like this, they're a boom and bust, cyclically abundant forage fish in the Gulf of Maine. We've seen them rise and fall over the years, and they're in an abundant period right now. So I do think keeping an eye on separating menhaden, Atlantic herring, and the river herring are all going to be really important angles now that we're able to get better taxonomic resolution in the diets. And maybe just the last thing I'll say is during the Amendment 10 scoping hearings, I think Don already kind of spoke to this, but the river herring conservation and advocacy was very strong. I think we share similar goals with the river herring conservation efforts. And so I think just anywhere where we can find allyship to provide information and look for those shared sort of like conservation alignment goals will be really helpful.
Yep. Yeah, I mean, it's fantastic that you're representing, the Northeast on those fisheries management teams and things, but I think we have a long way to go on the Mid-Atlantic and Southeast. Like, we don't even have good participation from the Southeast, or haven't a lot in AMBC. We didn't have a lot of samples collected from the Southeast for the Project Poop. So I think there's definitely room for improvement. And again, hopefully by working together, on these issues that might span the entire coast, we'll have a little bit more strength in what we're saying. So, maybe we can talk more about if you have specific recommendations on who in the Mid-Atlantic we should reach out to, or suggestions on how to do that.
Well, since it's a totally different management council, it's like a totally different suite of people and meetings and commitment levels. So yeah, I think we need to find somebody that has these shared interests down that way. And I think we've worked with a few folks in the past on like the sandland synthesis. I think it was Deirdre, but she may have moved on to another position. But anyway, I think they have some initiatives going in the Mid-Atlantic that also echo similar goals. So they have had this unmanaged forage fish series of goals, which are progressive compared to the New England Fishery Management Council, which doesn't have that yet. So I think they're thinking about similar issues. We just need to find a contact.
Yeah, they have a forage fish management plan, right, in the Mid-Atlantic. Yeah. Sorry, Ruth, I cut you off.
Oh, no worries. So I reached out to our fishery folks in the Virginia Marine Resources Commission, and I actually asked if they could have one of their staff become a member of the Atlantic Marine Bird Cooperative for this reason and for other reasons as well. And so they're going to get back to me. They have to get approval or something. But anyway, you know, reading these chats right now and, you know, seeing that they're already, menhaden are showing up in these New England colonies and they're fluctuating across years, I would think that fisheries managers would be really interested in the seabird information because it's just another piece of the puzzle as to the shifting distributions of the forage fish that are reflected in birds. And so, like, I'm really starting to get excited about this because Don, thank you for this whole idea of, you know, this could be sort of the foundation of a really, really cool proposal. And, you know, if we take the route of the competitive SWIG grants, and anyone who works for a state knows that they are just a nightmare to put together and to manage. But I think that, you know, if we get enough interest from this group, I could talk to our folks, our grants people, and, you know, see if they would be willing to have Virginia take the lead on this. Because yeah, this is just really amazing stuff, I think. And so, I mean, I feel like that this could be like a major next objective to kind of tackle. You know what I mean? And then Gemma, we could get money for you. And yeah, I mean, it could go, yeah, it's pretty cool. It could really.
Yeah. And I see your hand up, Doug. I was just going to say, not to volunteer Gemma for anything else, but like, it seems as if it would be super helpful for the Mid-Atlantic fisheries folks to hear that talk that Gemma just gave.
Yes.
To show them like, this is what is capable if you start collecting enough samples at colonies up and down the coast. Like, because we have been doing it for a number of years at a number of colonies in the Gulf of Maine, and now, you know, stretching into New York, we are able to start getting that amazing data that she just showed us. And so maybe there's an opportunity for something like that. Sorry, Gemma, again. But, you know, to demonstrate this is where we want to go, you know, we'd like to work with you on that process. How do we make this happen or something? But Doug, go ahead.
Hi, can you hear me?
Yes, we can.
Okay, great. Two things. I used to be on the regulatory discards committee of the, which is bycatch of the Atlantic States Marine Fish Commission, and they cover all the states. So we should have a bird representative if we don't. Usually it was someone high up in Fish and Wildlife Service that served on as the leaders of the group, the actual consul, but then any of the employees and stuff could be on the committees. On a second thing, I think that's really great that when you started talking about other species and that you wouldn't expect maybe that could be from discards of the ships, you know, fishing boats and stuff. And that can be a really good tie with the fishermen and the fisheries as to how so if you have birds that are just dependent on discards coming from cleaning boats, or, you know, I've seen trawlers with 10,000 gannets behind them. And, you know, so a lot of birds are out there dependent on the fisheries. So if you can, you know, just keep that in mind as something changes and you wouldn't expect it to be in the diet, that it's very well could be from the discards. And you could maybe use that as leverage. It sounds a little weird, but like artificial feeding to get them to discard in a way that might help the bird, you know, keep the stuff floating instead of discarding it below the water or something. But you should keep track of what fisheries are where and how that's coming. And Pat Jodas and Juliet did stuff on that with the terns and found them feeding mostly on shrimp coming from the discards of the trawlers in South Carolina or North Carolina, somewhere down there. Anyway, yeah, this is really great for that part. And who knows, you can make them as a mitigation. If you get in crises, have them discard in a different way.
Yeah, except in the Gulf of Maine, we'd like to reduce the lobster discards because it's usually the gulls that benefit from it. and then the gulls start turning. So you have to,
No, but I mean, if you know what the terns are feeding.
selectively, yeah, selectively who you're feeding. But it looks like Michelle has her hand up again.
Yeah, I have a process question. So on the suggestion of putting in a C-swig, there was a group of us that put in or started to work on a C-swig a few years ago. And we put a lot of work into the proposal, and then we couldn't find a state to be the lead. So I feel like it's worth having a conversation about making sure that we have a commitment for a state lead before people start working on a big proposal. And I see that some folks have also suggested other funders. But yeah, just learning from mistakes of the past.
Yeah, no, we actually, I think Virginia was going to be the lead on that as well. And then we ended up having to drop out because we just didn't have the capacity to do it. And that is such a huge issue with those grants. It is just, they are burdensome. But anyway, yeah, I think this warrants, a serious look into the feasibility of this and definitely exploring other funding sources. But maybe what we should do is get a group of folks together that has that's willing to tackle this issue and see if we can take it somewhere. I just think this is really exciting.
Yeah, it seems like, again, not to commit Gemma to anything, but it seems like that would be the next step, is if we want to include additional colonies or additional species, Gemma's going to need help to do that. Absolutely. And trying to get someone that could be, a real benefit to her in her lab seems critical. And so we need funding. And again, hopefully the fact that this year was so successful for all these new colonies, and then we have this amazing data set for the colonies that in the Northeast that we've been monitoring for a number of years, we can demonstrate the value of this project, and then also show that, in this very first year, look how well we did. Hopefully we'll just build on that. So
yeah. I was just looking at Carolyn Mostello's chat about seeing the waters boiling with Menhaden. So I just want to really mention a quick observation I made this summer, and that I too saw just massive schools of Menhaden in some of the in the Chesapeake Bay and some of the tribs, and amazingly there were no birds feeding on them, and they were, the little peanut size Menhaden, and they were definitely of good size. And it was probably in late July when the chicks were a little bit bigger, and it just amazed me that there were no birds working on those schools of fish, and they were right at the surface. It was just boiling with Menhaden. So it's all this is all really interesting stuff.
Yeah, we've seen that too, where you see a bunch of birds feeding. And then another day you see like 6 different areas with the water just looks like it's boiling and there's fish everywhere, and you don't see a single bird around. It's like, I try to convince myself that they're all full because they've already been eating so much all morning that they're just, can't eat anymore. That's what I like to think. So,
yeah.
So I don't know.
Yeah. Caroline, did you have a comment?
Yeah.
No, I was just saying that they're probably full. I mean, there's so many Menhaden, and I do see terns over them. I even see forsters, and but yeah, I mean, after a certain point, they probably won't be able to get off the surface of the water because there's so much to choose from. So they must need a break.
Yeah.
So we have about a half hour left. I have.
Well, can we just kind of, so I would like to at least see if there's a commitment from folks to possibly pursue this further. And also, Michelle, if you could offer guidance on how to pull in, folks from the fishery world into this as well. I mean, and we don't need to make any decisions right now, but does sort of everyone agree that this is something worth pursuing? And if so, you can just like raise your hand or do the hand raise thing. And yeah.
Yep.
Let's
And
see
I
if this will
know,
be worth it. I mean, it's going to be a lot of work for sure.
yeah,
Okay,
I
looks
definitely,
like,
it looks like there's definitely interest. And
yeah, thank
Michelle's
you.
on the chat that she had to run.
Okay.
I don't know where it was. I saw that somewhere, but so hopefully, Michelle did write sorry.
Oh,
Happy
good.
to help
Okay.
in funding pursuits. She has to run to another afternoon meeting, but she'll
Oh,
be
good.
she'll
Okay.
be here tomorrow. So it definitely seems like there's a lot of interest. And what I was going to say is maybe as a follow-up to this, we send out, an email to the larger group that says kind of with our next steps for the SCAW Forage Fish Working Group. And, this would be a priority to say like, okay, get back in touch with us if you want to participate, unless you're jotting down names right now, Ruth.
No, I don't recognize.
Let us know, and then we'll try to set up, another meeting after the holidays. What I was going to say is, like, I had slides that outlined the objectives of the Forage Fish Working Group and tried to show some of our accomplishments. I would rather have a discussion instead of me showing those. And the only other kind of thing I wanted to take away from this group or this discussion today was, if people thought it was worthwhile to try to do like a forage fish, like fact sheet to try to increase awareness and support of forage fish conservation, and do the same thing, like send out an email and, form a smaller group to try to work on that. And there's some really great examples. I had one from like the Teddy Roosevelt's conservation group that had like, this is what it looked. It was focused on bait fish and sports fishermen, but it was a very clear graphic that like without bait fish, then there's no good sport fish. And then here's all the implications versus like a healthy marine ecosystem. And I was like, oh, we could just substitute in seabirds and, things like striped bass to try to bring in other constituents. So I feel like those maybe
folks
would be
are,
like
how we can capture this interest by folks that have already started to do this is to, if you are interested in either helping pursue funding for, the diet Project Poop, put that in the chat, and then what Linda just described as well. And then we'll have that captured, and then we can get in touch with you.
Okay. And I'll try to pay attention here and write down some stuff. Looks like
Well,
Dawn.
it'll all be captured in the chat, so we can.
Oh, okay.
Catch that. Yeah. This is exciting.
Dawn, did you have a question?
No, sorry, just a legacy hand. Sorry about
that.
Great.
And one of the other things I had was like, are there other forage fish issues that are a concern to this group? Like, is there something that other people think we need to be focusing our attention on? I don't want to miss anything. Joan.
One of the things that I've been concerned about for several years has been the recovery of sand lance amidites in the cable trenches from offshore wind facilities. There's the general trend is to try and cable through sandy areas rather than rocky areas. It's a much better way to bury your cables. And there are sand lance in those sandy areas. And the question being, how long is the recovery time for some of these for amidites to recover in areas once the trench has been put in? And if you put together the sum total of, well, first of all, if you look at where some of the cables are going up Muskega Channel, within a half a mile of Great Gull Island. There are placements of several of the offshore wind cables because there isn't a central cable. There are many cables. Each wind farm has its own cable, right? So there isn't a central highway of cables. It won't be cabled once. It will be cabled for each farm. So my question is, how long does Amidites take to recover, if it recovers at all, in areas that have been trenched? And I think that at the scale that we were talking about offshore wind development, it could be notable. And in some sensitive places like Muskega Channel or like the area right off of Great Gull Island, it may be even more important. So that's something that has interested me and that I've talked to Vineyard Wind about. And they dropped a camera down while they were looking at their one of their export cable pathways and could actually see Amidites in the area. So you could actually do a before-after comparison in that. And when we look at, relative to forage fish, when I look at what our birds are eating out of Great Gull, if you take Amidites out of the diet, you probably don't get very many terns out the back end. So that's just something that I think is of interest to us at the scale that offshore wind is proposed. Is the recovery of Amidites in cabling trenches.
Thank you for that, Joan. And definitely that's something that we could bring up. It seems like there's offshore wind meetings like on a daily basis. And whether that's through like RWCS, identifying that as a priority, or I know Dave, I don't know if Dave Wiley's on the call or not, but Dave has coordinated a lot at Stellwagen Bank, a lot of sand lance research with folks at UConn. And so, they might be really good to bring into that discussion and look for guidance on how to best document the sand lance abundance, either during that process or after or something. So, yeah, that seems like a really good topic to focus on.
Yeah, I think that one of the questions is, there's a lot of discussion about the recovery around the turbines themselves, right? We've all sat in on a million of these meetings where we talk about how does the ground recover, what's the effect on the benthic community, but there isn't all that much on the cabling. There isn't all that.
Sorry, Liam Waters, who works at Stellwagen, has put in the chat that Stellwagen has a grant proposal out for a project looking at EMF radiation effects on sand lance and recovery times. So
yeah, that's great.
Sounds like maybe they're already on it, but maybe, I don't know if there's a way that we could, write letters of support and say that we feel like this is a priority project. You could let us know, Liam, if there's something that we could do to try to help support that project. Oh, go ahead, Liam.
Yeah, sorry. So yes, to answer or to expand on that, Joan, this project is focused on cabling rather than the turbines themselves. I'm not directly involved in the project, so I can't offer too much more of an elaboration on that personally. But if anybody is interested, I can get more information to you. Or if there's a better way of me just kind of providing that information to Linda or Caleb or anybody else to distribute more widely, I'm happy to do that as well.
Go ahead, Joan.
Yeah, I'd like to know, Liam, do you know who put it in?
It was, I don't know who the lead on it was. We had somebody on detail, Maggie Corey. I think that she did a lot of the work, but I know that Dave Wiley's name was on it, Tammy Silva, Pete DeCola. I can try to find it. It's probably somewhere in my inbox.
That'd be great. I'd like to hear more about it. I've talked to folks at Stellwagen about the same thing. I kind of beat this drum wherever I go. whenever we talk about fish and birds. That's great.
Good.
Yeah. That's great. Are there other forage fish issues that people want to bring up to the group, or any other concerns? The other question we had floated was, other guidance documents or other information that colony managers need. So back in probably 2019, I pulled together a bunch of diet monitoring guidance documents for terns in the Northeast. It was focused for Roseate terns, but it was applicable for common and Arctics as well. And we talked about fecal DNA and direct observations, and we kind of hit on using video cameras or game cameras a little bit, but we didn't have a lot of expertise at that time. Since then, there's been a couple of students that have done a lot of work, and so it seemed like it might be logical to update that document with better guidance on, best management practices or directions on how to use cameras, game cameras or video cameras to document diet. But we didn't know, I don't work with species like skimmers or gull-billed terns. I don't know if any of that information would be transferable to other species. But, is there information that maybe we could work together or share across this group that might help others start collecting additional information in the future on diet? But I don't know, maybe that's not a need and maybe everybody's good. We just wanted to try to kind of open this up for a discussion and see if there's other burning questions that people had.
Yeah.
Yvonne.
Yvonne, yeah.
Yeah, are the protocols to collect samples available somewhere? I'm thinking about other areas that would like to collect them as well, and maybe on different species or different sites, but so they could make it comparable to what's going on.
Yeah, so do you mean for the fecal DNA or the other diet sampling methods?
Sorry, no, the fecal DNA.
Yeah, I'm assuming Gemma's still on here, but she provided, like a one-page instructional sheet on how to collect the samples. So we could definitely provide that to other people.
Yeah, that could be on the website. That would be great, because I'm representing the Caribbean here too, and I think a lot of colleagues would at least know how it's done.
Doug.
Yeah, I was just thinking a little more about the bycatch and your sampling. It might be worthwhile when you're submitting samples, if you know what fisheries are going on around you, to put that in the ancillary data so that you might be able to make those links after you get weird results and things like that.
Right.
Yeah, that would be great. Good idea.
Yeah, I think, it'll be interesting to see once we get the results from, the fecal sample collection. I think that will probably drive sort of future efforts, I would imagine, including, the frequency of collecting samples. Does it need to be done annually? Can it be done every 3 years? That type of thing, right? I mean, as Gemma, as we get more results, they should be able to drive sort of kind of future sampling effort. And if we do need to explore other ways of collecting diet information, is that correct?
Yeah, and maybe, maybe we only collect from chicks. We don't bother collecting from adults because they're quite similar.
Right.
Thing. Yeah.
Yeah. I think one thing that most states expressed last year when we were organizing all this was that, that they would have the capacity to collect fecal samples. When you start getting into, collecting observational information, it gets tougher. Right. Yeah.
Yeah, because we kind of sold this as you potentially could have one day in the field and get your samples, where, you know, some of us have seabird technicians living on the islands for 10 to 14
Right.
weeks. And so
Right.
we have the capacity to do direct observations, but Gemma's work has shown that the fecal DNA analysis is still super important because we call it hake, but we don't know what, you know, we didn't know if it
Right.
was red, silver, or white. We're also misidentifying species. And then for us, it's been really helpful to identify what the adult diet is, because that was something we didn't know at all. But we felt like for the greater, you know, SCAW forage fish group here, that fecal DNA seemed like for those colony managers or researchers that had limited capacity, both money and time, that the fecal DNA would just be such a logical choice for so many reasons. And so that's why we were excited to get that project started this year. Yeah, are there other comments or questions? Everybody's disappearing on me. I don't know what's going on. Everybody's turning off their camera.
Sorry,
Will.
I had to get my dog.
Will, you had a question or a comment?
Yeah, what about how hard is it to import the samples? You know, if you're working in the Bahamas or in Turks and Caicos or something, how hard would it be to get those samples in? I imagine it'd be kind of a pain.
I'm going to pass that over to Gemma, because I know you've been collecting samples in other countries too. So
yeah, right now it's actually not that hard to get them in. I just need to send you our import permit, and you have to write a letter, which I'll send a template for. And yeah, it's not tricky. I don't know if there's any export restrictions in the Bahamas, but the import into the US is not that bad right now because they're considered differently from like avian blood samples, which are much more difficult.
So this is Doug. I'm a member of the Ornithological Council, and so on our website there are all the regulations as to import, export, and most of the countries are listed. And if you have any issues, or even if you need to know how that you don't find it on the website, you can call our executive director and she'll lead you through it. Plus she'll save your samples if you get stuck at customs and something gets screwed up.
oh, that's great. Thank you, Doug.
Thanks.
So we all pay for the Ornithological Council through our organizations that we belong to, Waterbird Society, Pacific Seabird Group, are members.
Good deal.
Yeah, that's great. I
Yeah.
didn't know that was
Thank you.
a thing. But it sounds like it shouldn't be that big of an issue, hopefully, for you to be able to get samples and submit them, Will. And I know was it last year or 2 years ago, we did have a master's student from Puerto Rico, right, that presented on fecal sampling
Right.
that he had done for his graduate work on a couple of colonies.
Yeah, I
So-
think he was here this morning,
Okay.
Lewis. I don't know if he's here now, but yeah. Yeah, and it's not actually that tricky to get samples
Yeah, I'm
into the
here.
US.
Okay.
Fine.
Great. Yeah. So there's already been, you know, some work started in that area. So that would be great to see it expanded.
So I'm wondering, can we take the last, what do we have, 10 minutes left?
Yeah.
And I'd like to pose a similar question to the group that that Linda did with regard to, you know, any priority issues involving colonies and breeding seabirds, right? And so we have the 5 focal species: common terns, black skimmers, least terns, double-crested cormorants, and laughing gulls. And just wanted to get a sense from you guys whether there was an interest in pursuing any sort of larger project involving either establishing productivity estimates across a broader range. One thing that I'm really interested in, because of the work that Virginia Tech is doing at one of our large seabird colonies, the Hampton Roads Bridge Tunnel, which I'm going to be presenting on tomorrow. But anyway, you know, just this large scale banding effort and having the ability to begin quantifying sort of the level of interchange of these birds among colonies, and, you know, which would include these birds showing up in colonies in other states and what have you. And, you know, that was one of the main reasons why we wanted to do this coordinated colonial waterbird survey, was so that we can get a count of, you know, these birds in the same year, so that we can get a much better population estimate, knowing that they do move around, but actually looking at a finer scale with uniquely banded individuals, I think would be pretty cool. And again, you know, we're all really busy, and we have limited funds and all that other stuff, but I was just wondering if there's sort of an interest in looking at more, you know, especially for these focal species, looking at these questions across a broader range. Diane?
Hey, thanks for all these amazing discussions and presentations. Ruth, yes, we're definitely interested in let's see, especially dispersal and connectivity across the different colony sites. We've got a fair number of chicks and some adults now that are banded from Poplar Island in the Chesapeake Bay. We've got, we're really interested in local dispersal, also interaction with what's happening with Hampton Roads and the changes down there. So within our region, but then also up the flyway. So we're definitely interested. We have been thinking about this for a bit more than a year and just trying to catch up on many other items, but I would love to hear from other folks as well. And we had some early discussions about this at the last Waterbird Society meeting as well.
Good deal.
Yeah. And before Lindsay asks her question or comments, I was just going to say, yesterday Ruth and I talked about this, and in the Gulf of Maine, there's a number of partners working on similar issues, and we have, you know, either metal field readable bands or colored field readable bands for roseate terns, Arctic terns, and Atlantic puffins, and are looking at kind of metapopulation dynamics. You know, a big part of that becomes data management. And so in the Gulf of Maine, we're sharing a database where we're all putting in the bands that we deploy, as well as the re-encounters of those birds, so that we can do analysis of it. And so, you know, obviously that's something important, because on that the data gets out of hand very quickly. But yeah, it'd be great. Lindsay, go ahead.
Yeah, kind of along those lines. Well, first of all, I mean, we're interested. I'm interested. Audubon, North Carolina has been banding Royal and Sandwich terns with field readable bands for a couple of years now, and plan to continue. But as we get into putting out more field readable bands, one of the really important things not only is to obviously not duplicate codes or colors and things like that, but to think about how to make those bands readable for people in the field. We also do a lot of reciting of these banded birds, black skimmers, royal terns, etc., and what letters or numbers you include, the size of the band, all of that. You've probably encountered that, you know, in the Gulf of Maine with things like puffins that I don't even know about. But I think it's really, really important to push people to think about that and put a lot of intention and thought into it, because when, you know, I'm trying to help the technicians, or I myself am reading bands, and we can't do it because, you know, of a design flaw basically in the way the band was made, that's really frustrating. And if you're in the field, you know, that time is precious. So being able to get that off the birds as easily as quickly as possible and make it easy for the public to do the same is really important. So I think there's a lot we can do with field readables and recitings, and I'm very supportive. And I also think it's really important to think about that aspect of it to make it as profitable as possible. That's my dead horse.
Spoken like a true banding database manager. The oyster catcher working group database.
Yes.
Thank you. Those are all good
Thanks, Terry.
points.
Yeah. We have about 5 minutes left. So I don't know if, again, people have any other comments or concerns. It seems like, we'll have a follow-up email asking for, I tried to jot down names from the chat of people who are interested in participating in the, in the grant effort to try to get additional fecal DNA funding. And then also want to reach out on who's interested in trying to do forage fish fact sheet or something.
Yeah, and can I just jump in real quick? And if there are folks who are interested in pursuing the whole idea of sort of a broad range banding effort, please put that in the chat as well, what we just talked about. It's probably not going to happen next year or the following year, but we can start talking about these things.
Go ahead, Tasha.
I just wanted to note, and if anyone's going to be a PSG or if you want to reach out because you're just interested in it, that I've been working in collaboration with Linda and with Keenan and with one of my students at Gettysburg to try to get some better idea of some of those larval fish distributions using GPS tagging data of the birds. So we've been linking GPS foraging points to the provisioning data of the birds to see kind of where they're capturing those herring and hake and everything. So just thought it might be of interest to this group. So make sure to check out Logan's poster if you make it to PSG, or if you're just interested in hearing more, feel free to reach out to us because I know getting to PSG is difficult for people this year. So just wanted to note that. Thank you.
Thanks.
Thanks.
Right.
Right. Maybe we're all talked out.
Yeah, just some of the other things that were on our list of objectives for SCAW involved, just for people to think about, managing human disturbance in sort of a coordinated fashion, with similar signage across, along the coast, having beach stewardship sort of programs along the coast. Another thing that's in one of our objectives is habitat enhancement, restoration, creation, all the usual stuff. And then also gaining political and public support for seabird conservation in general, which I think, some of your initiatives, Linda, certainly pointed that. So anyway, there's lots of stuff that needs to be done. Lots of things to think about.
Yeah, it looks like Yvonne has a question or a comment.
Yeah.
Yeah, Bruce, you mentioned earlier in your presentation the 2023-2024 waterbird survey. I was wondering if you could give an update on how that went.
Yeah, I think pretty well. So a lot of the work now on the colonial waterbird survey and also the database has been turned over to the conservation delivery specialist that's been hired also by the Atlantic Flyway Council. But as far as I know, all the states in the flyway, except for West Virginia, which they don't have many colonial waterbirds, did participate in the 2024 survey. There were some states like Virginia and Maryland where we were on board for doing a complete coastwide survey in 2023. So we did that and then followed up in 2024 by just collecting or just doing surveys on like double-crested cormorants and some of the other focal species just to add data into that 2024 effort. Because, well, I don't have enough time to explain the whole process, but basically it followed the sampling design. And, so the states at the 2023 survey were contributing to information to that sampling design that was actually developed by the Fish and Wildlife Service. So anyway, I think overall it went well. The best news, or the best thing that I think that came out of this is the fact that we've gotten all the states sort of dialed into doing these surveys, and hopefully with repeated encouragement, it becomes institutionalized in all state work plans to do surveys every 5 years. And so that's kind of the plan.
Okay,
or
great.
the dream. But I think it'll work. I mean, I think we got quite a bit of buy-in from states. So short of any catastrophe, lack of money, all those good things that you can't control, I think it's a go.
Great. Are there plans to I don't know, write some kind of report on it or something, like lessons learned?
Yeah, again, the data are getting entered into the AKN, so there'll be a test of the AKN as far as ease of pulling the information out and that sort of thing. Just haven't had time to go, but yes, there will be. And actually, the U.S. Fish and Wildlife Service is going to be pulling data here pretty soon because of the cormorant issue. They kind of jumped into the picture after the drafting of the 2020 EIS, cormorant EIS, where it mandated that good population estimates were going to help dictate the amount of take by state. So permitted take of cormorants. So they'll be generating something fast, I think. So
yeah.
All right. Well, thank you so much, everybody, for I think a really productive discussion.
Yes.
And hopefully you'll be hearing from Ruth and I soon about follow-up actions. We have a 10-minute break scheduled, and then the Marine Spatial Planning Working Group is going to meet from 2:55 to 4:55, and that's the link that you used for the session this morning. You're going to go back to that link. So again,
Right.
Thank you so much. Thank you, Gemma, too, for that amazing presentation. And yeah, looking forward to hearing the rest of the talks tomorrow and on Thursday. So thank you all for participating.
Yes, thank you so much, everybody. Take care and all your input. She left. Stop it.